It's very tempting to want to think of pyruvate dehydrogenase as a single enzyme that catalyzes the transition step, but that is not the case. The pyruvate dehydrogenase is a massive complex filled with many, many enzymes. So in fact, it's more of a factory than it is a single enzyme. In fact, it's the largest known multi-enzyme complex. If you look at the molecular weight, it's 9.5 million Daltons. So this thing is astronomical. And within the complex, there are many copies of 3 totally different enzymes with totally different jobs. The very first one is pyruvate dehydrogenase, also simply known as E1. And it's at this complex, we'll soon see, that in fact the pyruvate loses a carbon dioxide and the 2-carbon group from that pyruvate is transferred onto a temporary holder, and we'll see more of that action soon. You'll probably recognize what happens at E2, which is dihydrolipoamide acetyltransferase. You guessed it. This is going to be in charge of the transfer of the acetyl group, and in fact form very much fits function for this enzyme, which has an elephant trunk on it and is able to move from one active site to another with that acetyl group. It's actually lastly at E3, which is sometimes also called the E3 binding protein in literature, but this is the dehydrogenase, and this is the actual location where we see the generation of reducing power. So if we try to envision 3 different enzymes all present in multiple copies, it's hard to even wrap one's mind around that. But we can kind of think about the idea that they're all working in concert with one another. It's really symphonic. And the E2, there's 60 copies of E2, and it's at the center of the enzyme. And in fact, it's arranged in like this pentagonal dodecahedron, where at each of 20 vertices, there's 3 different copies of E2. So add that up, that's 60. And so at the center, we get the E2. And then also kind of clustered in the kivas where E2 isn't in the center, we see E3. And then like overarchingly like a shell surrounding everything else, we get the E1. And I have a picture of this actually published in 2001 in the Proceedings of the National Academy of Science, PNAS. And it's interesting because this is just going to be mind-boggling to see, so you should actually probably dim your lights wherever you are so that you can get the like true impact and power of this multi-enzyme complex. Yeah, right? And notice there's E2 with its sort of elephant trunk structure. It's in the green. E3 tucked away in the kivas in the red. And then finally E1 forming that shell around it. So it is mind-bogglingly large. This particular image, you're going to look at it and think, I've never seen anything quite like that. Well, that's because this was generated using something called cryo-EM. This is really just a high-resolution electron microscopy at very, very low temperatures, just as you might guess. But people began after 2001 wanting to try to grow a crystal of this, and in fact probably many were started before that, and they couldn't do it. They simply cannot grow a crystal of this pyruvate dehydrogenase complex. And so last I heard, they were actually hoping that they might be able to do it in outer space. Yeah, grow a crystal, that is. And so I couldn't find, I combed through the literature on both the web of knowledge on the library's database, but also on Google Scholar, and I can't find any evidence that they've successfully crystallized it in outer space. If somebody finds that article, extra credit, send it to me. I would love to know if they had succeeded in that. What I did find was the role that the pyruvate dehydrogenase complex plays in cancerous cells. Cancerous cells tend to have upregulated glycolytic pathways and downregulated aerobic respiration. And so if you can upregulate the pyruvate dehydrogenase complex, then what will occur is that will be inhibitory to a cancerous cell. And so because of that, they're looking to try to target enzymes that would upregulate, and we'll learn more about these enzymes soon, that would upregulate the pyruvate dehydrogenase complex and encourage aerobic respiration, right? Encourage further oxidation of pyruvate. So this is a very, very recent study, 2016 March of this year. So at this point, we're ready to dive into the incredibly complex mechanism, tracing what happens at each one of the three enzymes, E1, E2, and E3.
Gear up for more fireworks as we realize that this reaction mechanism not only requires enzyme 1, enzyme 2, and enzyme 3 in this multi-enzyme complex, but it also requires co-substrates and prosthetic groups both of those enzymes. So let's look at the mechanism, and as we go through it, let's pay special attention to which are prosthetic groups and which are co-substrates, and how that makes a difference in the roles that they play. Starting out then with enzyme 1, it's going to be a prosthetic group of enzyme 1 that will play the first role. This prosthetic group is called thiamine pyrophosphate, or TPP. TPP can actually ionize, losing a proton and forming a carbanion that you see right here. And in this ionized form, it's actually termed Ylid. But this TPP that can become Ylid is the prosthetic group of enzyme 1. So the larger outer yellow regions of the complex that we just looked at has this particular prosthetic group. So prosthetic group of E1. Now this Ylid, this carbanion of Ylid, is really going to be the thing that sets everything in motion. Because remember, our overall goal is to oxidatively decarboxylate the substrate pyruvate. Ylid is going to start that process by actually acting as a nucleophile, attacking the electrophilic carbonyl carbon of pyruvate. That's going to cause a couple of things to happen. First off, the CO2 will act as a leaving group, and there you've got the first goal, the loss of one thoroughly worn out, fully used up carbon dioxide. So that product is formed very rapidly within the mechanism. So that's our first step, if you will, as pyruvate dehydrogenase attacks with its prosthetic group at the carbonyl carbon. So that gives off a carbon dioxide, and one product of the reaction has now been formed. But remember, we have two more products yet to form, also acetyl-CoA, right, and NADH. So the rest of these steps will be all about forming those other two products. Notice now that the acetyl group, or what will eventually be the acetyl group, the 2-carbon group, has been transferred on to the prosthetic group of E1, and it forms hydroxyethylthiamine pyrophosphate. So see, that 2-carbon moiety has now been linked onto the prosthetic group of E1. But remember, this is a prosthetic group, right? So it's not like it can run off and somehow transfer this 2-carbon group onto coenzyme A or something like that. It can't. It has to stay at the enzyme active site, because that's what prosthetic groups do. They don't move, right? They're the in-house waiters. So what will happen then is that instead of it moving to transfer the 2-carbon group, enzyme 2 will actually come to the heart of this. And it turns out, this is pretty cool, it turns out that enzyme 2 actually has on the enzyme what I sort of think as an elephant trunk that's actually movable. So this inner, what we saw as the green inside of the multi-enzyme complex, it actually has this sort of elephant trunk on it, and it can actually move into the heart of enzyme 1. And that's what's going to happen next. The prosthetic group, go ahead and write that down, HETPP. So next, the prosthetic group of enzyme 2 on the elephant trunk of enzyme 2 is going to make its way into the active site of enzyme 1. So this particular prosthetic group of enzyme 2 is actually called lipoamide. So lipoamide is going to be attached to a flexible arm on enzyme 2 that's going to come, you know, kind of snaking in to the active site of enzyme 1. And at this point then it's going to react with the HETPP, and the 2 carbon group is going to be transferred from HETPP to lipoamide, forming dihydrolipoamide. So essentially what we're going to get there then is the addition of an acetyl group onto the prosthetic group of enzyme 2. And of course now Y-lid is reformed, and that's good because we know that's true of prosthetic groups, right? They can be momentarily changed during the process, but they cannot be, you know, they have to return to their original state before the end of the reaction can occur, and so that looks good. Now everything is at all the action now is at enzyme 2. So enzyme 2 now has gained the 2-carbon acetyl group. We've still only seen one product of the reaction formed, and that is the CO2. We see evidence of an acetyl group here, but no coenzyme A yet, and that's what's going to happen next. So at this point, acetyl dihydrolipoamide has been formed. So we've seen basically the coupled oxidation of HETPP to the reduction of lipoamide and the concomitant transfer of a 2-carbon acetyl group onto that enzyme 2 prosthetic group. Now remember that enzyme 2 has this flexible sort of elephant trunk on it, so it's simply going to swing over now to its own active site. So it's just going to swing into the E2 active site, and it turns out that coenzyme A is a co-substrate of enzyme 2, so it's there ready and waiting for its 2-carbon acetyl group. So E2 swinging into the E2 active site, so E2 swinging in the elephant trunk of E2 swinging into its own active site, and that's where it finds its co-substrate coenzyme A. Notice the dihydrolipoamide, or the lipoamide as it began, is a prosthetic group. It needs to go from its original form and back within the course of the reaction, and it's not going to dissociate from the enzyme. Contrast that to the co-substrate acetyl-CoA, or what will become acetyl-CoA, the coenzyme A. Contrast that. Remember, co-substrates are the pizza delivery people, so they're going to be able to disassociate from the heart of the enzyme and take off. And that's important because we know that coenzyme A is going to need to deliver the acetyl group to the TCA cycle. So we'll see then the acetyl group right here getting transferred from the dihydrolipoamide to coenzyme A, forming acetyl-CoA, and here we have it, number 2 product of all of this. So you might start thinking, well, aren't we done here? I mean, we've got the CO2's been given off, the acetyl-CoA, it's done, it can go to the TCA cycle, right? Aren't we done? Don't forget, though, the other product. Remember, this is an oxidation, and don't forget that NADH is another product, an important product of the transition step. So we have to look at how that is formed, and also, hey, note this, dihydrolipoamide has not yet been converted back to lipoamide, and we know that since it's a prosthetic group, it has to be before this reaction is considered complete. So we'll see then that the rest of the action is going to be in the oxidation of dihydrolipoamide, and that's actually going to occur at enzyme 3. So now this flexible elephant trunk of enzyme 2 is going to swing into the enzyme 3 active site, where it is going to be oxidized. So here is the dihydrolipoamide swinging into the E3 active site, And at the E3 active site, you might think, oh, I bet that's where Na plus is, ready to take on the electrons and become NADH. Well, sort of, because what actually happens is a little more convoluted than that. First, dihydrolipoamide is oxidized, transferring electrons to FAD. So the prosthetic group of E3, and it's going to turn out that E3 has both a prosthetic group and a co-substrate. So the E3 prosthetic group is FAD. It's going to get reduced by dihydrolipoamide, and we're going to form first FADH2. So of course E2 is the enzyme that's catalyzing this, even though it is at the heart of E3. So now we have accomplished one of the goals that we set out to accomplish in this step, and that is that the lipoamide prosthetic group of E2 has now been completely converted back to where it started. So that's one goal down, but yet we still have not released to the NADH that we know is also a product of the transition step. So that means that the FADH2 prosthetic group is going to have to transfer electrons to NADH, and that's what happens next. So NAD plus will gain the electrons from FADH2. NAD plus is the co-substrate of E3, so note, E3 has both a prosthetic group and a co-substrate. So uptown, huh? An in-house waiter and a pizza delivery person. So this NAD plus takes on the electrons and becomes NADH. And it's E3 that catalyzes that reaction. The cycle is now complete, right? We now have generated all three products that we know we needed to in this process. We have generated the thoroughly worn out, all used up carbon dioxide, generated the acetyl-CoA that is also product, and finally the NADH. So the last product to be formed is actually the reducing power. Nice.